My name is Kevin Su. I'm a Ph.D. candidate in the program for experimental and molecular medicine here at Dartmouth, and I'm a Superfund trainee. My work focuses on investigating arsenic-contaminated drinking water and its effects on the immune system. No, I don't have a private well. All my drinking water comes from the town of Hanover, so a public water source. It has been shown in studies that many private wells, private drinking water sources that are not under federal regulation have high levels of arsenic in their drinking water. I definitely recommend testing your drinking water. Arsenic exposure is correlated with an increased risk of a variety of diseases such as cancer, cardiovascular disease, and skin lesions. Also, with an increased incidence of viral respiratory infections and as well as greater morbidity in response to viral respiratory infections, which is what we in the NO lab are focusing on. So a better understanding of the discrete mechanisms which arsenic affects would definitely help us find a solution for remediation. I believe that many scientific publications as well as organizations have done a good job of conveying to the public the risks associated with arsenic exposure. However, there are still many individuals in the United States as well as abroad that are exposed to high levels of arsenic, particularly in their drinking water, so I definitely believe there's more work to be done. One of the powerful tools that we use in the lab to investigate the effects of arsenic ingestion on the immune response to viral respiratory disease is the use of mouse models. So we have mice that are chronically exposed to arsenic from their drinking water, and then subsequently we infect them with the flu. And from those mice, we harvest biological samples that give us very informative data about the effects of arsenic on the immune response to a viral respiratory disease challenge. This tool is very powerful because it's in a living system which can give us a very good representation of what we might see in people. One of the procedures that we do is an ELISA assay, which is a method in which we can colorimetrically determine the concentration of a certain protein that we obtain from a certain biological sample. In terms of our studies, we primarily look at the concentration of albumin in the airway of mice after a flu infection. This lets us determine the measure of lung injury that these mice have, so we can compare with mice that were given arsenic and mice that were not, see differences in lung injury. We can investigate and observe those samples that we collect, investigate those immune cells, see what has been changed due to arsenic exposure. Ever since I was young, I was always impressed by what scientists have been able to accomplish in regards to developing treatments, cures, or even further understanding of diseases. That impression and sense of awe has carried with me all the way until now, and because of that, I'm very happy to be part of biomedical research. What I enjoy the most is directly applying what I've learned from the graduate program in terms of methodology and approaching scientific problems and using that to actually help solve real-world problems. I enjoy coming into work every day knowing that I am potentially finding something new, discovering something new that has a global impact on the health of millions of people, and being part of a research group that is involved in that feels great.